Good morning everyone. Welcome back to Biology with a Difference with your instructor Babita Rani. In the last class we studied about the nucleus and the structures that are present inside the nucleus. Today we are going to study other organelles of an eukaryotic cell. What we are going to study today is about the endomembrane system. All of you know that membrane bound organelles are only present in eukaryotic cell and each organelle has their function, but there are certain organelles which work in coordination and they are known as endomembrane system. They constitute the endomembrane system. What are those different organelles which constitute the endomembrane system? It is the lysosomes, the vacuoles, the endoplasmic reticulum and the vacuole. So, what are endomembrane system? Endomembrane systems are set of membrane bound organelle in an eukaryotic cell that work in a coordinated manner. And what are the four organelles which constitute this endomembrane system? Number one is endoplasmic reticulum. Number two, Golgi bodies. Number three, lysosomes. And number four, vacuoles. So, these are four organelles which constitute the endomembrane system. They work in coordination. We will see how each of them are coordinating each other's work. And the four organelles are endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. Today we will study in details the endoplasmic reticulum. Endoplasmic reticulum was first of all observed by Garnier, and the term endoplasmic reticulum was given by Porter. We generally write endoplasmic reticulum as ER. So, from now we will write ER for endoplasmic reticulum. Endoplasmic reticulum is of two different types. One is smooth endoplasmic reticulum SER and the other one is rough endoplasmic reticulum RER. So, endoplasmic reticulum was first of all observed by Garnier and the term endoplasmic reticulum was given by Porter. Its ultrastructure was seen by Porter and Claude, and there are two different types of endoplasmic reticulum depending upon the presence and absence of ribosomes. One is smooth endoplasmic reticulum where ribosomes are not present. Other is rough endoplasmic reticulum where endoplasmic reticulum is studded with ribosomes. Now, if we look at What is endoplasmic reticulum? The parts of endoplasmic reticulum, it has three different parts: cisternae, vesicles and tubules. This part is mainly present in RER and this is present in SER. Tubules are present in both of them. Let us see what is the function of, if ribosome is present on RER, the function of ribosome is to synthesize protein. So on RER, protein synthesis takes place. One function of RER is to synthesize protein. So protein synthesis takes place on RER. If we look at SER, lipid synthesis takes place on as you know, lipid and protein are important constituent of cell membrane. Cell membrane is made up of lipid and protein, and both of them are manufactured in endoplasmic reticulum. Protein on rough endoplasmic reticulum and lipid on smooth endoplasmic reticulum. So one function of endoplasmic reticulum is to help in biogenesis of cell membrane or living membrane. Bio means living, genesis means formation. So one function of endoplasmic reticulum is to help in synthesis of cell membrane, and it is known as biogenesis of cell membrane. If lipid is produced at SER, where do you think it can be present? It will be present in those areas where more lipid is there. So it is present in adipocyte. There we store fat. Adipocyte. Where is adipocyte present? It is present in the skin, which stores the fat. It is present in liver cells. It is also present in cells which secrete steroidal hormones like follicular cell in case of female, and Leydig cell in case of male. So we have, these are the places where more SER is present. Now if ribosome synthesizes, RER synthesizes protein, this is the site of protein synthesis. So where do you think RER may be present? This is present more in islet of Langerhans, that is a part of pancreas and brain cells. Do you remember the Nissl granules of brain cells? That has more of present in cytone. Nissl's granules that is present in cytone, that is basically RER. So these are some of the places where RER or SER may be present. Let us differentiate the RER and SER on the basis of their function and location. Let us see first the location location. It is present in pancreas. More of it is present in pancreas and brain. More is present in liver cell, adipocyte, follicular cells, and Leydig cell. What will it synthesize? Because ribosome is present here, it helps in protein synthesis, or protein is synthesized here. What will be synthesized here? Lipid synthesis takes place here. Another function of SER is detoxification of drugs in liver. That is why this is present more in liver cell. So detoxification of some drugs takes place in liver. This is another function of SER. One more function of SER is glucogenolysis takes place in SER. So let us see some common functions of endoplasmic reticulum, that is general function of endoplasmic reticulum. One is it provides skeletal framework to the cell. As you see, if this is nucleus and this is cytoplasm, it is present in between the nucleus and cytoplasm, and it is connecting both of them. So it forms a skeletal framework for the cell, and as it is connecting both the ends, other function of it can also be, it helps in transport of substances in the cell. From nucleus to the cytoplasm can also take place through ER, and from cytoplasm to different organelles can also take place through the endomembrane system. The third function, if we consider a common function, is biogenesis. As I talked earlier, biogenesis of cell membrane, because both lipid and protein is synthesized here. The fourth function that we can have is formation of cell plate. Formation of cell plate takes place in plants. Cell plate during cytokinesis of plant cell in cell division. So these are some of the functions of endoplasmic reticulum. It provides skeletal framework to the cell. It helps in transport of substances in the cell, as it is connecting link between the cell membrane and the nuclear membrane. It helps in biogenesis of cell membrane, as it is both protein and lipid is being synthesized in the endoplasmic reticulum, and cell membrane is composed of lipids and proteins. It also helps in formation of cell plate, that is during cell division in the process of cytokinesis. In cell division, after karyokinesis, cytokinesis takes place when the cell has to be divided into two different parts. So here what happens, during cytokinesis in plant cell, cell plate formation takes place with the help of endoplasmic reticulum. So these are some of the common functions of endoplasmic reticulum. what was the difference between the endoplasmic reticulum, smooth endoplasmic reticulum and rough endoplasmic reticulum. If we look at it once again, rough endoplasmic reticulum is present in pancreas and brain. Smooth endoplasmic reticulum is present in liver cell, adipocyte, follicular cells and Leydig cells. Rough endoplasmic reticulum helps in protein synthesis, whereas smooth endoplasmic helps in lipid synthesis, and it also helps in detoxification of drugs, and that takes place in the liver. One thing that you have to remember here is endoplasmic reticulum is present in all the eukaryotic cells except mature mammalian RBC and germinal cells. So this you have to remember, that it is present in all eukaryotic cell, but it is absent in mature mammalian RBC and germinal cells. So this was all for today in endoplasmic reticulum. Tomorrow we will study lysosomes and Golgi bodies. Thank you for watching this video. Please do subscribe and click on the bell icon to get the notification whenever a new video is released. If you like the video, do share it with your friends. Thank you for watching once again.